In this short video, we will look at transduction, the use of viruses called bacteriophage to move DNA between bacteria. Bacteriophage, and here is an image of one to my left, are viruses that infect bacterial cells. The bacteriophage has two different life cycles, lytic or lysogenic, but a bacteriophage can go through both. In the life cycle, the bacteriophage attaches to the bacteria and injects its DNA. The DNA is then transcribed. The so-called early genes are transcribed first, and these make proteins that can make more of the phage. Next, the late genes are transcribed to make the virus capsid. This is the virus coat. And finally, the enzyme lysozyme is transcribed, which causes the bacteria to lyse and release the virus so that it can go on and infect other nearby bacteria. In the lytic's life cycle, there is an ordered series of events leading to the final lysis of the bacterium. However, in the lysogenic cycle, the phage DNA is incorporated into the bacterial chromosome by site-specific homologous recombination. The bacteriophage can revert to being a lytic phage if required. The phage DNA that has been incorporated into the bacterial chromosome can be recognized for repackaging into phage by the presence of Pack sites. However, during packaging, some bacterial DNA may also be captured, and some phages, for example P22, do not correctly recognize the Pack sites and therefore package host DNA, which can then be passed on to the next bacteria the phage infects. This also means that the phage can be used for gene mapping through the capture of bacterial DNA. However, there is one issue in that the head of the phage has a limited amount of space and so therefore cannot contain a lot of DNA. This limit is called the head-full rule. Interestingly, besides being used for the transfer of DNA between bacteria in the wild, phage do have some commercial uses, such as killing unwanted bacteria on food, detecting the presence of bacteria, and they may also have a role as a replacement for antibiotics or for disarming bioweapons.